Hello everyone, I'm Diana Karaki, PhD student in my last year as a Paris Clay. Today's presentation is about combined MS and MSMS deconvolution of SWATH DIA data with the DIA-NMF workflow for comprehensive annotation in metabolomics. What is metabolomics? It's the study of endogenous and exogenous small molecules in biological systems, known as metabolites, and nowadays is a key tool for biomarker discovery. Liquid chromatography coupled to high-resolution mass spectrometry is a reference technology in this field. However, identification is still challenging due to the diversity of compound structures and the limited size of databases. For this, tandem mass spectrometry enables to provide identifications by acquiring the fragmentation spectra MS or MS2. And modern instruments nowadays enable to combine for every MS1 scan several MS2 acquisitions using different protocols. We are interested in a SWATH DIA protocol, which consists of the biological sample is first injected through the liquid chromatographic column. At this stage, molecules are separated by retention time according to their physiochemical properties. At each time scan, existing compounds are ionized before entering the first mass spectrometer, where it measures their mass to charge ratio to form the MS1 spectrum. SWATH DIA fragmentation protocol, it separates the full range of the MS1 spectrum into overlapping continuous isolation windows. And each isolation window, all precursor ions exist in each isolation window are fragmented using a collision energy before entering the stacked mass analyzer to form the MS2 acquisition. Using this protocol, we are sure that we don't lose any information regarding any metabolite exists in the biological sample. However, the MS2 spectra are hybrid mix. They contain mixture of fragments related to co-eluting and co-isolated MS1 ions. For this, the main challenge remains, which is the unmixing in order to achieve the correct identifications. The state-of-art existing SWATH DIA processing pipelines, untargeted pipelines, are MSDL and commit DIA, which are based on model peak selection, which is the traditional way of deconvolution for such kind of pipelines. After detecting peaks and constructing the mixtures, they select model peaks from these mixtures, and the elution profiles are decomposed as a linear combination model of these peaks. However, to form the MS2 pure spectra. However, one limitation of this model peak approach is the chosen model peaks may be not optimal. They can be related to two different but two co-eluting compounds. And the researchers in 2019 in ADAPT GC4, they used the multi-curve resolution, MCR, NMF approach, and they show that it outperforms the traditional way of deconvolution, specifically for co-eluting compounds. As you can see here, that the scores of L-histidine and L-histidine were improved after using the MCR. So, as a first step in my thesis, we were motivated to use the NMF, which is a blind source separation, to deconvolute the SWATH DIA data. It's never used before on such kind of data. And you can see, and we implemented this classical pipeline. We didn't use the model peak selection, but we used an NMF approach to achieve the deconvolution. You can see the implementation and our results in our accepted paper in the Signal Processing Conference, SIPCO, in 2024. But what is NMF? We have a non-negative matrix X. In our application, it consists of sources mixed up in unknown but a linear way. And our aim to factorize it, to approximate it by finding two non-negative matrices A and S, where S contains the basis of the matrix X, in our case is the pure spectra, and A contains the elution profiles, the coefficients of these bases. For this, we adapted and we modified the NGMCA NMF algorithm, and we solved this non-convex optimization problem. We divided into two sub-problems, convex problems, and each one is solved iteratively until convergence using the forward-backward step algorithm. There is a key fact why NMF work in such kind on this SWATH DIA data, which is all parent ions and their fragments have identical elution profiles in MS1 and MS2. Using our first version of the workflow and the state-of-art existing pipelines, they process the peaks peak by peak, which introduce a lot of redundancy and time consuming. And our core goal is to solve the challenge of the generalization of the untargeted case by comprehensive unmixing of entire file while unlocking the full potential of NMF on wider retention time ranges and in both levels, MS1 and MS2. For this, we thought, why we don't apply NMF on a retention time windows, which have all MS1 and MS2 signals. For this, we developed our workflow, which is called DIA-NMF or DIA-NMF. It's a framework for untargeted compound identification. So we detect the MS1 peaks from the raw data using XCMS. Then, at each iteration, we select a retention time window. For each time window, we extract all MS1 plus MS2 data. We align them before concatenation, and we apply the NMF deconvolution. Sorry. After each deconvolution, from each isolation time window, we have all the pure sources for all existing metabolites or compounds in this window. We apply some post-processing filters to delete noisy or unmeaningful biological sources. In order to evaluate the performance, we used a real biological sample. It's a human plasma spiked with 47 known compounds at 8 different concentrations. And for each concentration, we have 3 replicates. And for sure, it was processed using SWATH DIA. For every MS1 scan, we have 10 MS2 acquisitions covering it, and the isolation window sizes ranges between 20 and 50 Dalton. And for spectral matching, we use the fragment presence product and the reverse dot product and the fragment presence ratio. Go rapidly to the results. The DIA-NMF is the only example which provide pure MS1 spectra. And also, comparing the identification rate between the DIA-NMF in light green, in blue for MSDL, and in orange for the commit DIA, the DIA-NMF outperforms in terms of the identification rate in all workflows, in all files, especially at low concentrations. In more details, our MS1 spectrum, the pure MS1 spectra, group all related ion species, adducts, isotopes, and neutral losses. And the MS2 and the recovered MS2 spectra integrate fragments originating from all relevant SWATH isolation window. And for a general view, you can see here is highlighted by a star the maximum mean score of the 2 scores for each compound sample combination among the 3 different workflows. And it shows here that the DIA-NMF extracts spectra of higher quality for a lot of the compounds. The last result I want to show it that the DIA-NMF successfully unmixing coeluting compounds in terms of retention time plus masses. For example, in this retention time window, we have 2 endogenous compounds of the same precursor but elute at different times. After deconvolution or unmixing, sources 3 and 4 were assigned to these 2 coeluting compounds. And after matching their pure spectra with the MS1 and the MS2 databases, they achieve higher scores, which means that the DIA-NMF successfully unmix all compounds exist in each retention time window, even if they are coeluting. So as a conclusion, here we introduced our DIA-NMF untargeted SWATH DIA processing workflow, which is more rigorous and efficient because of the simultaneous deconvolution of both MS1 plus MS DIA data. In it, we introduced a new deconvolution strategy that extracts all pure components from each retention time window in a single step, limiting redundancy and decreasing the running time. It outperforms existing references tool in terms of identification rate and spectral quality. And the most important, it was implemented and is freely available R package. Just yesterday we launched it, so I will be happy to know your feedbacks about it. And we also implemented a lot of functions and features to export, to plot, and visualize the processing before and after the deconvolution to make things easier for chemists. As a perspectives, we are working on enhancing the MS1 data extractions to recover all signals from the raw data, even if the algorithm used, such as XTMS, didn't detect them. We are able to identify them. We are developing multi-sample unmixing strategies, and we will apply it to biomarkers discovery through case and control comparative analysis. And thank you for your attention.
Thank you. We can do one quick question.
Thanks. I'm curious, like for your method, does it apply to the DDA experiment as well? Or like for the DDA, the co-eluting is not a severe issue?
Yeah. No, it can be applied to DDA for sure, because it's much easier than DIA. But yes, it can be. But now the R package that we just launched, it works just for deconvoluting MS1 data and plus DIA data, but it can be done easily.
All right, thank you.
Thank you.